comes to moving through membranes, most proteins can't just freely move through, but they also can't just call an Uber. Thankfully, there are uber cool ways that we can get molecules a ride to the other side. So today, let's talk about membrane transport.
If you think about a membrane, the first thing we probably think about is that lipid component. And numbers wise, there are more lipids than other things. But mass wise, about half of the mass of some of our membranes actually comes from proteins. And about a quarter of all of our protein coding genes encode for membrane proteins. There are a huge variety of membrane proteins, and they play all sorts of important roles. These include serving as receptors so they can respond to extracellular signals. And then some serve as signal relayers, so kind of like they can work with receptors to respond to those signals. They can also act as like scaffolds to hold things together in the membrane or hold membrane proteins kind of in place, keep them from diffusing too far. They can also form as cell-cell and cell surface adhesion molecules. We're not going to get into those functions today. We'll get a little more into signal relay later in the course, but today we're going to talk about transporters. So these are membranes that are actually going to help things get through that membrane. So again, there's all sorts of different types of proteins, and we're not going to get into all of them. This picture from your book kind of gives a little overview. What we're going to be talking about mainly today are going to be what we call these integral membrane proteins that actually go through the membrane. So some proteins only are attached to like one side. We call these peripheral membrane proteins, but the integral membrane proteins, these are at least like they go somewhat through the membrane, and the ones that we're going to talk about are actually going to go all the way through the membrane. One of the important things to think about when it comes to membrane proteins is that the transmembrane part, so the part that actually goes through the membrane, is kind of inside out. So we talked a lot about how normally proteins kind of have their hydrophilic parts on the outside and their hydrophobic parts on the inside, away from the water. Well, this is true for the parts of membrane proteins that are exposed to the water, such as the endodomain, so the part that's like inside the cell, and the ectodomain, the parts that's outside the cell. But as for the transmembrane domain, the part that's actually going through the membrane, well, here on the outside, it has to have those kind of hydrophobic parts, and on the inside, it'll have those hydrophilic parts. And if it has like a channel or something like this, if it doesn't have a channel, then you'll basically just see that all of it's going to be hydrophobic in the middle. But when it comes to serving as these roles to transport things through membranes, here you're typically going to have some sort of hydrophilic part to provide an alternative route through the membrane for things that normally wouldn't be able to get through because they're hydrophilic. As to how these proteins actually get through that membrane, like the transporter proteins, they can do this typically as either alpha helices or beta strands and sheets. So going back to our protein structure, what level of structure are we talking about if we're talking about helices, strands, and sheets? Right, that's going to be our secondary structure. So remember with an alpha helix, we kind of had this helical shape, and it's kind of more compact. Whereas with a beta strand, things are kind of more spread out, and we have that alternating up, down, up, down, up, down, so that every other amino acid is facing the same face. As a result, if we have a beta strand going through a membrane, basically at least every other amino acid is going to have to be hydrophobic. And then you might have every other be hydrophilic if it's part of a pore, or if it is not part of a pore, then you might have them all be hydrophobic. Because it's kind of stretched out, it only takes about 7 amino acids per pass. So these membrane proteins can have like multiple passes, so they can go through many times, or just like a single pass, where it just goes through one time. But if we're talking about a channel, then we have things with multiple passes. In terms of alpha helical passes, well, here, because it's kind of more compact, it takes about 20 to 30 amino acids per pass. And it's common to have about 7 or so passes. Between the pass segments, as well as like at the exposed ends, so these parts that are kind of like sticking out and at the very ends, these are going to be hydrophilic as well. And these might also play a role for anchoring other things. And depending on how many passes you have, these ends might be on the inside or on the outside. And there are tons of different types of proteins and ways we can classify them based on where those ends are, but we're not going to get into that. Just know that typically you're going to have your transmembrane domains be alpha helices or beta barrels or sheets. So basically beta barrels are where you kind of have these sheets that are in these circles. And those are going to be a common way. You have a pore that's kind of ringed by these beta barrels. These beta barrels themselves aren't the actual pores, but actually having like a ring of these beta barrels will form a pore that things can get through. And these are just a few examples of them. So now that we know how these membrane proteins can get through the membrane, let's talk about how they can help other things get through the membrane. Let's talk about how membrane proteins can serve as transporters or channels and pores and things like this to help molecules get through membranes. This help can be in the form of what we call active transport, where it like requires energy such as ATP, or the coupling to a favorable transport, or it could be passive, no energy required, just a selective pore or something, or even just simple diffusion. And things like this can actually serve as providing energy for our active transport, which is what we call secondary active transport. And we'll get into all of these concepts. But first, let's think about what would make it favorable or unfavorable for something to move through a membrane. Basically, the molecular motivation to move comes from gradients. There are a few different types of gradients that we could talk about. We can refer to chemical gradients, in which case we have things moving from high concentration to low concentration. And this is just happening basically via diffusion. So when you dissolve a solute, basically you're going to start with where you dissolve that solute, you're going to have a high concentration of the solute molecules. Those molecules are going to move around randomly. And based on the randomness of their movement, they're going to kind of end up dispersing themselves. And then this is where the movement is ultimately going to lead to the net movement from high concentration to low concentration, where you would then ultimately end up with an even distribution. There's also electrical gradients. So just as we've been talking about how we have all those like IMFs and things like this, everything from molecular interactions to when we're doing an electrophoretic gel. We're moving from, in the case of the gel, we're going from negative to positive, but you can also go from positive to negative. Bottom line, opposites attract, like charges repel, and this can serve as motivation to move. So we have the combination of both the chemical gradient and, in the case of a charged molecule, the electrical gradient, which together give us what we call electrochemical gradients. So here is an example. I'm kind of showing what's going on inside and outside of our cells in terms of membrane potential. So this is going to be electric gradient from electric gradients. And then when we're talking about concentrations, this is referring to our chemical gradient. And you don't need to know these exact numbers, but what you can see is that basically inside of our cell compared to outside of our cell, is sodium going to be higher inside of our cell or lower inside of our cell? Yeah, sodium is going to be lower. So we have lower sodium. And then what about our potassium? Yeah, our potassium is going to be higher. But then outside of our cell, we're going to have the opposite. We're going to have higher sodium and lower potassium. I like to think about special K. Our cells are special. And so inside of our cells, we have a high concentration of potassium. If we were to just think in terms of their chemical gradient, which way would that potassium want to go? Yeah, the potassium would want to go out and the sodium would want to go in. And if we think about in terms of our electric gradient, where would they want to go? Basically, inside of our cell, what this is saying when it says a membrane potential of minus 50 to minus 70 millivolts, this is saying that inside the cell is more negative. And so if inside of our cell is more negative, well, then they both, just based on the charge alone, want to go in. So obviously we need to consider both the electric and the chemical gradients, but then we also need to think about, wait a second, we just said that if all things were being even, if everything could kind of move freely, there'd eventually be no gradients because you would be diffusing out to an even concentration. You would be having those positive charges go to the minus charges, neutralizing everything, making that so that that membrane didn't have any membrane potential. So clearly, if we have a situation like this, there's something going on. Our cells are actually working to keep things out of balance. And so today, that's what we're going to talk about. So basically, instead of just letting everything through, membranes are what we call semi-permeable. So by permeable, we're talking about allowing things through, and semi-partially. So basically, this is partially permeable. The things that can get through, we say that they are penetrating. We can have like a penetrating solute. So in this example, basically this solute A is going to be penetrating. So the membrane is permeable to solute A as well as to the water. Both of these can get through the membrane. However, it's impermeable with respect to solute B. So we say that solute B is going to be non-penetrating. So if something can't get through the membrane, we say it's non-penetrating. If it can get through the membrane, we say it's penetrating. And then if we think about things from the membrane's perspective, it would be permeable to things that are penetrating and impermeable to things that are non-penetrating. So those are just a couple of the terms that we can use to describe what can and can't get through a membrane. And so now let's talk about why things can and can't get through membranes. Some membranes are only freely permeable, so through like simple diffusion, so things just moving through on their own. Only a few things can get through there, such as like smallish nonpolar molecules, because remember we've got that the lipids are mostly hydrophobic, even though they've got those hydrophilic heads, as well as small polar but neutral molecules. So this includes water. So water also has channels called aquaporins to help more go in and out, but water can also diffuse to some extent through that membrane. And I know it sounds a little weird because we're like, wait, lipids are hydrophobic and we're talking about how like lipids repel water and all this stuff, but it turns out that water's small enough that a little bit of it can get, that some of it can just diffuse through. But if we're talking about something charged, even if it's really small, that's not going through. And if we're talking about something bigger but non-polar, but polar, such as like a sugar maybe, that's going to need help getting through as well. So in order to get those other molecules through cells, through the membrane, we need help from integral membrane proteins, or IMPs. And cells can then use energy to move molecules in order to maintain those gradients, like we saw in the case of our sodium and potassium ions, and having them in concentrations that would not happen if we had things just move to equilibrium, where everything would end up being even. And so we're going to see how we can use energy, we can use various transporters, we can use all these different strategies in order to maintain gradients and maintain what we call like potential. So basically, separated gradients is going to lead to potential energy. And we could talk about chemical potential, electrical potential, and osmotic potential. Basically, if we think back to like our example of being in a roller coaster and being at the top of a roller coaster and kind of having like potential energy, you have the potential to go down the slope and release energy. Similarly, if we have, we can have like chemical or electrical potential, osmotic potential, basically potential energy that comes based on having being able to kind of move from a region that's less favorable to a region that's more favorable, and therefore having a thermodynamically favorable reaction that's going to be able to release energy. So there are three main types of potential: our chemical potential. So this again is just going to depend on the concentration of that specific molecule. So each molecule is going to have a chemical potential that's in terms of that molecule's concentration inside and outside of the cell, or inside and outside of a membrane-bound compartment. We also have the electrical potential. So sometimes this is abbreviated Bm. This is going to depend on the concentration of all of our charged molecules. So this depends on all of the ions, not just the specific one we're looking at. We can always talk about like an electrochemical gradient, but basically it's only going to matter if the molecule that we're moving is going to be charged. And so we'll see how we can see an equation. And if we don't have a charged molecule, then this part of the equation where we're dealing with the electrical potential will kind of just cancel out and equal zero. And that's going to depend on all the molecules as opposed to just the one you're looking at. Another gradient, another potential that I just want to mention, but that's not going to be specific for our molecule moving, is actually when our molecule can't move or can't move well, and basically instead it's the water moving. So the osmotic potential, this deals with the movement of our solvent. So in the case of biochemistry, our solvent is going to be our water, and it's going to depend on the concentration of all the solutes in a mixture. And this is what we refer to as one of those colligative properties. So going back to our chemistry, there's colligative properties, things like our freezing point depression, those things that depend only on the concentration of the dissolved particles. So in the case of our osmotic potential, basically what we're saying is that our solute is non-penetrating, so the membrane is impermeable to it, but water is penetrating, water can go through. And therefore, what's going to happen is that this water is going to go through and kind of like dilute out the solutes. This is not really how it's happening. Basically, it's just that random movement of water, and kind of if there's more other stuff around, the effective concentration of water is going to be less. So it's like there's less water inside, like there's less water when there's a lot of solutes. And so then the water is going to move from its high concentration to its low concentration. So you're going to get this net movement from the high water concentration to the low water concentration. But then this is also going to be movement from the low solute concentration to the high solute concentration. But you have to think about things in terms of water, and then you can see that it's really going from our high to our low. We're not really going to talk about osmosis here, but remember that going back to our idea about why we wouldn't want to store sugars as like for storage as monosaccharides, because then basically you'd have high solute concentrations that would drive a bunch of water into the cells. Well, that was coming because the cells aren't freely permeable to those solutes, but that doesn't mean that solutes don't have a way to get through cells. And so now let's talk about those ways. Basically, the way that we're going to take is going to depend on whether or not it's kind of energetically favorable to go into a cell or to go out of a cell. And when we're talking about energetic favorability, when we're talking about our thermodynamic favorability, we need to think back to our good old delta G. And so remember that a negative delta G is going to be thermodynamically favorable. This is going to say that our end state, so if our molecule's in the new place, it's going to be happier than if it was in the old place. And whereas a positive delta G, this is going to be unfavorable. Basically, we're saying that we're taking something from where it likes to be to somewhere it likes to be less. And so how do we know where it likes to be more or less? Basically, we can calculate this free energy change for transport, this delta GT. I know we've seen like a bazillion delta Gs, but this is going to be the delta GT for a molecule. Is going to be equal to RT times the natural log of C2 over C1. So basically, this is saying your concentration of place 2. And then this is going to be your concentration at place 1. So your initial, the concentration at the new place divided by the concentration at the old place, and then plus this part. So basically this is telling you about the chemical potential. And this part is telling you about the electrical potential. And so overall, this is going to tell you about the electrochemical potential. So if you have an ion, if you have a charged molecule, then what's going to happen is this electrical part is going to be important. The Z is going to be the charge of that ion. F is just a constant. And this is going to be the transmembrane electrical potential in volts. So basically that is going to be like our VM. And basically, so that's going to be determined for the membrane, dependent on all of the ions inside and out. The F is going to be a constant, and basically your Z, that's the thing that's going to change depending on the charge of the molecule. Now, what this means is if we want something to be energetically favorable, we want a negative delta G. We can get a negative delta G if basically what happens is we have that the concentration of our at the new place is less than the concentration at the other place, or if we have that the electrical potential is going to be favorable. So it's kind of like the combination of the two that's going to make things matter. If something has a negative delta G, so we'll do some example calculations in class in our exercises. So don't worry about those for now, but we will do those to see that basically what we're going to find is that if we have a negative delta G, if things are favorable, our movement is going to be exergonic, so it's going to produce energy, and therefore it can move via what we call passive transport. So it doesn't need energy in order to get it to move. Instead, we're going to get energy if it moves naturally. Conversely, if we have something with a positive delta G, therefore the movement is going to be endergonic, it's going to require energy, and therefore in order to get it to move, we're going to have to put in some work, we're going to have to use active transport. So here it moves kind of because it wants to, and here it moves because we're kind of forcing it to. So there's going to be different ways that we can kind of allow things through a membrane. These are going to be broken up into active transport and passive transport. So basically, this side of things is going to be passive, and this is going to be active. If we're talking about passive transport, this is going to be along its gradient. It's going to be in the favorable direction with regards to its potential. We don't need to put in energy. But if it's against the gradient, it's against that potential, then we need to use active transport. We're going to actually have to put in energy. And so now let's look at different ways in which we can do this. First in the overview, and then we'll go into some specific examples. So with passive transport, remember that this is along our gradient. So this is going to be in the exergonic direction. So it's going to be energetically favorable. We mentioned how there were a few things that could kind of freely diffuse. So nonpolar molecules and really small polar ones. And they're going to be able to diffuse simply based on their electrochemical gradient. So it's going to be an electrochemical gradient, but really it's just going to be a chemical gradient. And why? Well, because we can't get ions to diffuse freely. Just can't diffuse freely through the membrane. So in our equation, basically that Z is going to be 0, and so that part's going to cancel out. But I like to kind of just think about things in terms of the electrochemical gradient, to use that same equation, and just have that part not matter. Okay, so but we can have our nonpolar molecules and our really small polar molecules just diffuse through. Some things are going to need help to get through, and so like our ions, those that we were saying couldn't just diffuse through, but still they can have a favorable inside versus outside. They can have a favorable drive to get in. They just have a barrier to actually getting through that membrane. If we can provide them a channel, if we can provide them a route through that membrane, that's going to give them some hydrophilic stuff that's going to make their transport easier, then we can allow them to diffuse along their gradient and still be energetically favorable. And so we can do this through like selective channels. If, however, we want to move things in the direction that is unfavorable thermodynamically, well, here we're going to have to put in some energy. There are two main forms to do this. We can call these primary active transport and secondary active transport. Again, we're going to get into examples of each of these and a little more of the details. But with primary active transport, here you're directly coupling the movements to an energy producing chemical reaction. So this is going to be like hydrolysis of ATP. It could be like you actually phosphorylate the transporter itself, and this causes a conformational change, or you kind of just burn the ATP, and this will cause a conformational change without actually phosphorylating the protein itself. But this is somehow going to couple that movement directly to that that energy producing chemical reaction. Whereas with secondary active transport, here basically we're using energy to transport one molecule, and then since we then make a favorable gradient of that one molecule, now we're able to use that drive from the energy that's released when that molecule goes along its gradient to drive something against its gradient. So here, basically, we're swapping one molecule for another. One is going to be in the favorable direction, and this is going to power the movement of the other in the unfavorable direction. Which gets me to the point that basically some of the proteins are going to let through multiple substrates, and some of them are only going to let through a single substrate. Uniport, these are going to be single. So uniport transporter is going to be a single substrate. And then if we talk about multiple, then we call this co-transport. And this can be symport, in which they're going in the same direction. So in this example, they're both going in. Or it can be antiport, in which they go in opposite directions. So one goes in and one goes out. And it doesn't just have to be one-to-one. So we'll see key examples, like one of the ways in which we're able to maintain that gradient, in which we have sodium higher outside of our cells and potassium higher inside of our cells. We'll look later at how one of the key ways that we actually maintain this gradient is through this sodium-potassium ADPase. This is an example of an antiporter, so it's letting one thing out and letting another thing in. Well, actually, in this case, it's letting 3 sodiums out and letting 2 potassiums in. In this way, we're able to kind of pump out the sodium, pump in the potassium, maintain the potassium concentrations higher inside of the cells, and those sodium concentrations higher outside of the cells, providing that chemical gradient that would have disappeared if you just let things go freely through. And we'll see that basically we're going to use energy from ATP in order to do this, because we're having to move things against the gradient in order to maintain the gradients. And we're also going to have to, in the case of the sodium, be moving something positively charged from a negatively charged environment to a positively charged environment, which is against its electrical gradient. But first, before we get to the active transport, let's go back to our passive transport. So our simple diffusion, remember here, we don't have any channel required because the molecules are freely permeable. So this can be like really small polar molecules, like our water, or it can be non-polar molecules, but not our charged molecules. For charged molecules, for things that are bigger, things that are more polar, basically we're going to need facilitated diffusion, which is going to use selective channels to make the journey through the membrane easier. So the reason why most things can't just freely diffuse through the membrane is because they're facing a high barrier to actually doing so. Now, when we're talking about these channels and these transporters, we're not really talking about enzymes, but we can still consider kind of like an energy of activation, this delta G double dagger. And so that's what's shown in this figure. Basically, it's going to have a high energetic cost in order to get through this membrane in the absence of a transporter. And why this is basically we have to think about, okay, we have this molecule outside and it's going to be surrounded by water, so it's hydrated. Now we have to say, okay, give up your interactions with the water and now make interactions with this hydrophobic lipid. And the molecule's like, dude, no way. Like, I'm a hydrophilic molecule. I was happy out here in the cytoplasm. I'm all polar, hanging out with the water. I'm not going to give up my bonds to the water in order to form interactions with these lipids, which aren't going to offer me much. And so you have to strip the water off and then go through this membrane that you don't really like And then you can get back and you can get your water again. But going through that lipidy stuff, that's going to be a huge barrier. And so what's going to happen is basically, if you have a protein that provides a channel that's going to offer alternative bonds, it's going to offer things to kind of make up for the bonds that you lost to the water. It's going to provide a more hydrophilic passage. This is going to lower that activation energy for the transport and allow your molecule to get through. These channels can be highly selective because you can imagine you don't just want huge pores in your membrane that let everything through, or else that wouldn't be very helpful. So instead, these channels are highly selective. There's different strategies that they can use to be selective. This is an example of a potassium channel. So we have a lot of different channels and pumps and things like this for dealing with potassium and sodium. This is just one example. And in this example, what you can see is basically that when the potassium is outside, it's going to be hydrated. So it's going to be connected to these water molecules. Now, as it goes in, it's kind of going to replace the bonds to the water molecules with bonds to the backbone carbonyl oxygens in this channel. And this is going to help it get through the membrane with still favorable reactions. And then once it gets out, it can kind of rehydrate. Somewhat interestingly is that sodium is smaller than potassium, but sodium can't get through this channel because sodium, when it's hydrated, it's going to be too big. And when it's not hydrated, then basically it's too small to form these favorable interactions. And so this channel is going to be selective for potassium, even though you would think like sodium should be able to get through. So cells can be really clever. Proteins are awesome. Often passage through these channels is going to be regulated. So instead of just having a channel that's open all the time and therefore would allow things to kind of diffuse to reach their equilibrium, which remember would deflate all of our potentials and make it so that things wouldn't have any drive to get through. Instead, we can regulate the passage through channels. There are a couple of ways that this can be done. Some of these are kind of like have a single gate, so they're basically closed on one side, and then you can open that one side. Some of them are kind of going to be more like pumps, and they're going to have like alternating gates where one gate will open to one side, and then you have something happen that causes that gate to close and the gate to the other side to open. So different strategies for regulating passage in and out. An example of a single gate is going to be like a gated ion channel. These are going to allow for rapid flux of inorganic ions in response to a stimulus. So basically, the stimulus can be something like the binding of a ligand, which we call a ligand-gated ion channel, or a change in voltage, so a voltage-gated ion channel. So both of those stimuluses, there will be different ones. Some will respond to the ligand and some will respond to the charge. But these are going to both be ways in which you can kind of open up the gate and allow these molecules to get through quickly. Here is an example of a ligand-gated channel. This is going to be a nicotinic acetylcholine receptor, and it's going to be permeable to both sodium and potassium. What you can see here is basically it has these multiple subunits, like a lot of membrane proteins, like a lot of transporter proteins. Up here, you have the acetylcholine binding site, so that's going to be our ligand. When that binds, what's going to happen is that basically the channel is going to open. So initially, this channel is kind of closed, and you can have things like come in, but they can't get through. It's going to be open to the outside. Things can come in, but they can't get through to the inside. Then what's going to happen is that the acetylcholine is going to bind. This is going to change the shape. It's going to open that gate, open that channel, allow these ions to flow through. And this is going to help your nerves communicate. Another way that we see nerves communicating and things like this is with charge. So we have voltage-gated ion channels. Here, a change in voltage is going to cause conformational changes that open that pore. And so how could you get a protein to respond to a charge? Well, we have to think about, well, proteins can have charges too. So at the termini of proteins, as well as with amino acids that are either basic or acidic, basically they're going to respond to charge. So positively charged things are going to get attracted to a negative charge, and they're going to get repelled from a positive charge, and vice versa for our negative charges. And therefore, we can have these proteins undergo conformational changes in response to an electrical stimulus, like our nerves send out. And so this is an example of a potassium channel that's going to be opened in response to voltage. So in the closed form, you can see that there's not really space for this potassium, but in the open form, once it's gotten that electrical stimulus, now you have space for that potassium ion to go through. So those were the examples, though, where we're going along the gradient. We are going in the favorable direction potential-wise. But now let's talk about when we have this unhappy situation where we actually are trying to take something from where it's happy to somewhere where it's less happy. We're pushing our ball up the hill. We need some energy to do so. And there are a couple of ways we can do this, which is primary active transport or secondary active transport. So our primary active transporters are also known as like pumps. And so they often have these two gates, and one is going to open to one side. Things bind, and then you open to the other side. And a few main types are going to be ABC transporters, P-type ATPases, and V and F-type ATPases. You don't need to worry about these V and F-type ATPases for this class. Basically, these are a family of ATP-dependent transporters that transport protons. The ones that I do want you to know about are going to be these P-type ATPases and these ABC transporters. And so we'll go into each of them in a little more detail. There's a wide variety of ABC transporters, and they pump a wide variety of different substrates. So some of these are really selective, and a lot of them are not so selective. But some of the things that they let through are going to be many drugs, so like pharmaceutical drugs, as well as things like amino acids. And they're doing this across a membrane against a gradient. In this example, on this illustration, basically they're showing a channel that's going from the extracellular space to the cytoplasm. But we also see examples when we're going from the cytoplasm to the extracellular space, pumping, say, drugs out of cells and contributing to drug resistance. But for this example, basically they have the extracellular space out here and the cytoplasm in here. What these family of transporters, one of the things they have in common is kind of their structure. They have these two transmembrane domains, so going through the membrane, as well as inside or on one side, you have these two ATP binding domains, sometimes called cassettes. And so basically ATP binding cassettes. And then this is going to basically bind to ATP and cause a conformational change upon the hydrolysis of that ATP that's going to lead to the substrate being moved through. So you start with the channel open in one direction. ATP gets hydrolyzed, so the substrate can come in. ATP gets hydrolyzed. This leads to those transmembrane domains undergoing a conformational change that's going to open the channel to the other side and let the substrate out. The ADPs can then be exchanged for new ATPs, and you could have those be hydrolyzed and have this cycle occur again. There are a lot of different ABC transporters, and you don't need to know their names. Because a lot of you are interested in medicine and things like this, I thought I would mention a couple medically relevant ones. We have this ABCB1, and basically this is going to be a multi-drug resistance protein that's important for meeting drug resistance. So sometimes cells will become resistant to various drugs because they kind of just pump those drugs out of the cells. And this ABC transporter is fairly non-selective, and so it'll pump a lot of things out. And so this can be a big problem. Another thing that can be a problem is our ABCC7. And this is our CFTR, or it's a chlorine channel. And basically it's going to cause defects in it, cause cystic fibrosis. And so there's a lot of work that's done on that. And that's going to be an example of an ABC transporter. The other type of transporter that I want you to know about is going to be these P-type ATPases. These are a family of cation transporters. So what are they transporting? Right, they're going to be transporting positively charged ions. And so some of the main ones that we'll think about are going to be calcium, sodium, and potassium. And we have examples of pumps of the P-type ATPase family that are going to do all of these. And so, for example, one that we'll look at in more detail is going to be this sarcoplasmic endoplasmic reticulum, calcium 2 plus ATPase, or the CIRCA pump. And basically what it is it's a uniporter for calcium. So what does it mean if it's a uniporter? Right, it's only going to be transporting that one thing through. And so in this case, it's going to be transporting calcium through. Inside of our cells, we kind of like store calcium inside of intracellular compartments so that we can release it really quickly to have signaling effects. And so the CIRCA pump is one way that we can do this, and we'll take a look at how it works in a minute. This is another common example is our sodium potassium ATPase. This is the one I was telling you about earlier, where it helps maintain that gradient of where we have the sodium higher outside and the potassium lower outside and the potassium higher inside and the sodium lower inside. This is one of the ways that we can do this is by pumping out 3 sodium ions for 2 potassium ions. And so what type of transporter would this be? So what type of co-transport is this? Yeah, it's anti-port because basically we're taking one thing out and swapping it for one thing that's going in. And in this case, we're transporting both of them against the gradient, which is why we're going to have to couple it to the use of ATP. What these families of P-type ATPases have in common is they happen to have the same structure and way that they're actually going to use that ATP to phosphorylate the receptor, to phosphorylate the pump itself, and then this will cause the change. So instead of just burning ATP, instead of just splitting that ATP, we're actually going to phosphorylate the pump. So here's the example that we'll look at in a little more detail, this CIRCA pump. So remember what it's going to be doing is it's kind of going to be taking calcium and pumping it into that compartment. And in order to do this, it's going to undergo these shape changes. Now on the left, it's a little confusing because this is like the open form, but basically what it's saying is that it's open to calcium. So you have this channel open so that calcium and ATP can come and bind. So the calcium is going to come in through this channel and kind of bind up here, and the ATP is going to bind down here. Basically, though, what's going to happen is that then this channel is going to open to the outside or to the lumen, so to the inside of that vesicle, and allow the calcium through. And so let's look at a little more detail about how this is happening. So basically, you have this transmembrane domain that the calcium is going to help the calcium go through the membrane. But then you also have these 3 intracellular domains, A, P, and N. You start from having the transmembrane domain have a channel that's open to the inside. So remember when we thought about like our 2 gates, this is going to be one of our gates. And we think about like the inside and the outside, it's a little confusing. Just think about like this side versus this side. And so basically the calcium is able to go in. Once it binds, now you also have the ATP binds, and ATP is going to bind down here. The binding of ATP is going to kind of cause this N domain to move, and it's going to cause the P domain to get phosphorylated. So this is going to get phosphorylated using the ATP. So as opposed to the ABC transporters, here we're actually phosphorylating a crucial residue inside of our protein itself, and that's going to cause further shape change. So you get this change that's going to cause conformational changes, that's going to release the calcium on the other side. So it opens the other gate. So you basically enter, close the one gate, then you use ATP to phosphorylate the domain, then you open the other gate, and now you just have to reset things. So it actually releases the ADP, dephosphorylates itself, and resets so that you can do this again and again. So those were examples of primary active transport where we're directly coupling the use of an energy generating reaction, typically the use of ATP as we saw, either directly by phosphorylating the protein or kind of just by hydrolyzing the ATP itself. And this was going to allow us to provide the energy to get the molecule to move against its gradient, or molecules. So remember, we could have co-transport, in which case we're transporting multiple molecules, or we could have uniport, where we're transporting just a single one. Finally, let's talk about secondary active transport. Here we're going to couple the movement of a molecule against its gradient, so requiring E, pushing that ball up the hill, to the movement of another molecule along its gradient, so producing energy. So if we think back to our kind of roller coaster, the energy that you get going down one hill is going to get you up the next hill. That's the idea that we have with our secondary active transport. So somewhere along the line, we're going to have to put in that energy to get that first thing up the hill, but then we'll use the movement of that first thing down its hill to cause the movement of the other thing up its hill. So we're not really going to talk about that many examples of this in particular, but this is going to be one example, the sodium glucose symporter. So what does it mean by a symporter? Right, they're going to be traveling in the same direction. So what's going to happen is basically you'll have this transporter that's going to allow sodium and glucose to come through at the same time. You're going to be going along the gradient for sodium. So remember that sodium is going to have a higher concentration outside than inside, and so it's going to be happy to come inside. What's going to happen is that the sodium is then going to come inside the cell, and then this is going to be coupled to the movement of glucose, which is going to be energetically unfavored. And so because you have this high extracellular sodium, this is going to drive the movement of glucose into the cell. These secondary transporters will typically, what they'll have is basically they'll have sites so that, and they'll be regulated in a way that they're only going to open when both of those things are bound, so that you don't have it. You're not wasting the movement of the sodium without actually having glucose there to go through. And then remember that the reason why we have this sodium have the high concentration outside is because we're actively pumping it outside, such as with our sodium potassium ATPase that we're using to maintain that gradient that we can use to drive the movement of glucose into the cell. So a lot of different things. Just remember that basically if things are moving against the gradient, we're going to have to put in some energy to do so. This can be primary, where we're directly using ATP or a similar molecule. We're coupling directly to a energy generating reaction to change the shape of a channel and let things in or out. Or it could be secondary transport, where we're coupling the movement of one thing along its gradient to the movement of another thing against its gradient. Sometimes, however, we don't need to put in energy to get things to move because they already have potential. So that electrochemical potential that comes just from having differences in their concentrations inside or outside, and or differences in the charge inside or outside that can then provide them the energy that they need in order to move through the membrane. However, there's actually not enough energy for them to move through the membrane if it weren't for most of these, if it weren't for having specific channels in which to do so. Because moving through that membrane is going to require swapping out all those bonds to water for bonds for like passage through a lipid that is not going to offer favorable things to a hydrophilic molecule. Instead, what's going to happen is basically these channel proteins are going to provide a hydrophilic route through. They're going to provide interactions that replace the interactions that are lost to water and allow these molecules through. By having the molecules like not be able to freely diffuse, but to only be able to go through certain channels, you're able to regulate what goes in and out. These channels can be highly selective, and they can also be regulated by things like ligand binding or things like voltage. In these ways, the cells can allow things to go energetically favorable directions only kind of on demand when you want them to. And then there are those few rare things that can kind of diffuse through, those nonpolar things, those really small polar molecules like water, which can also go through aquaporins and stuff, but simple diffusion is what we call it, when these molecules can kind of just go through the membrane itself. But you're not going to be able to take ions through the membrane that way. You're not going to be able to take bigger things, not going to be able to take bigger polar things. Those things are going to need help. And then again, we can have our passive transport, where basically we just provide a way through so that they can travel along their gradient, or we do a more active transport, where we're actually having to use some energy either directly, which would be primary active transport, or indirectly, which would be secondary active transport.